Hello and welcome to this webinar titled Big Data Strategies for Cell Therapy Manufacturing, hosted by Biopharma Asia magazine and presented by Scott R. Berger, MD, Principal of Advanced Cell and Gene Therapy at LLC. This presentation will be followed by data capture and analysis through the cell therapy value chain, presented by Heidi Hagen, co-founder and Chief Strategy Officer for Vineti, Inc. My name is Stephen Edwards and I'll be your moderator. Before we begin, I would like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the questions tab located directly below your webinar screen. We'll go through these at the end of the webinar. Now please allow me to introduce our first presenter, the Principal of Advanced Cell and Gene Therapy, a consulting firm specializing in cell and gene therapy product development, manufacturing, and regulatory affairs. Dr. Berger has over 25 years of experience developing cell and gene therapy products and has consulted for over 100 industry and academic clients in North America, Europe, Asia, and Australia. He has directed or consulted on process development, manufacturing, and regulatory aspects of a wide range of cell therapy and gene therapy products, including CAR T cells, gene-edited cells, dendritic cell vaccines, natural killer cells, and other immunotherapies, hematopoietic stem cells from bone marrow, blood, umbilical cord blood, and regenerative medicine products such as MSCs, chondrite cells, and other somatic cell therapies. I will now be handing over to our first presenter. Welcome, Scott.
Thank you very much for that gracious introduction, and thank you for the opportunity to speak here today. I wanted to discuss big data strategies in cell therapy and gene therapy a bit as we are beginning the commercial era in cell therapy. And so as we advance from relatively small, more academic clinical trials or relatively small industry clinical trials and begin really trying to treat the large numbers of patients who are potentially in need of the kinds of therapies we deliver. We have the opportunity to generate very large quantities, we will generate very large quantities of data, and we have the opportunity to learn quite a lot from them. And so with that, a bit about how we might approach big data. Well, first off, let's consider some of the problems that the data-related problems that we face in cell therapy and gene therapy. Our manufacturing processes tend to be quite complex and in general are not adequately understood, or perhaps to put it more kindly, are not well understood, not well defined. Critical process parameters, those parameters that we need to monitor and control in order to assure that the process is in good control and will result in the product that we want. Critical process parameters need to be identified and controlled, and identifying critical process parameters, CPPs, is not an entirely simple matter. Currently, we identify CPPs in a manner that is, well, slightly better than best guess. This is what we think is important about the process, but it takes a great deal of experience with the manufacturing process and a great deal of process data to tease out really what are the critical parameters of the process that we need to measure and to control. Manufacturing processes also tend not to have been optimized. We've generally improved them to the point where we can run them and expect them to be reasonably reliable, but all the same, we frequently end up with manufacturing processes that can fail for reasons that are difficult to predict. That's one thing if we're treating, say, 50 patients in a phase 2B trial or something, but it's quite another if we're trying to treat thousands of patients a year as we hope to do in the commercialization phase of many cell therapy products. This lack of optimization extends to to purely practical matters like cost of the manufacturing process, the yield of cells that we get. Our cell yield at the end of most cell therapy manufacturing processes really is the cells that we generate are generally worth more than gold, especially since you can get gold, but nobody else can make the cells that are the result of your manufacturing process. So that's really what everything depends on. Operational efficiencies. It's one thing to be as cost effective as you can be if you're doing a 300 patient clinical trial or 10,000 patients a year. Our supply chains and cold chains are often inadequately controlled and inadequately documented, and this is everything from collection of cells that are the starting point for our manufacturing process to shipping of the cells that are the final product, but also the supply chain for non-living raw materials, for viral vectors or for cytokines or media and so forth. All of these complexities need to be accounted for and optimized. Our products are also very complex and are also inadequately understood. It's common for us to say the manufacturing process defines the products, and this is a common regulatory, not admonition, but a regulatory approach. This is absolutely correct and understandable. But the fact is that process defines the product is a necessary approach because it reflects analytical limitations. If we can more thoroughly understand the critical quality attributes, the critical characteristics of our products that relate to function, that relate to efficacy, then we'll be able to define our products more fully. And although the process will probably always define the product for something as complex as a living biologic product like a cell therapy, well, we should be able to define the products more extensively by testing, if we understand its critical quality attributes and how to test for them. Current understanding of critical quality attributes, even very late in development and into commercialization, is often very much a legacy of early stage R&D, and indeed the product may have critical quality attributes that we simply have not fully appreciated early in development. Product testing also is generally not optimized. We're oftentimes using analytical methods that very much grow out of an R&D environment. This is true for identity and purity, but especially for potency. Simply defining the relevant biological functions that underpin potency can be a considerable challenge and requires a great deal of data supporting it. These are things that big data can help us with. So, given some of what I would consider the data-related problems that I've just described, what are some of the data-based or data-driven opportunities? Well, I mentioned before that most cell and gene therapy products tend to have somewhat limited data sets, even by the time they reach pivotal trial or commercialization. Now, we simply haven't had the opportunity to run the manufacturing process, if it's an autologous product, thousands of times. If it's an allogeneic product, we simply haven't had the opportunity to see what our banked allogeneic products, how that performs in thousands of patients. So there's quite a lot of opportunity to learn as development of the product progresses, and especially as we get to commercialization. And by opportunities, I'm thinking substantially of process-related data mining and analytics. At present, what we do in the way of data mining and analytics is could more be thought of as little data, not big data, simply based on N alone and based on the analytical approaches, the data analysis approaches that we take. In fact, most processing technology that we use in our manufacturing processes tend to capture relatively little in the way of processed data, less generally than the analytical technology that's used in biotech or in protein bioprocessing, or for that matter, even basic things like Google Timeline. If you simply think about the example of Google as and the way in which it gathers masses of data, some of which is obviously useful, some of which might be useful at a later date. If you think about it that way, and apply that way of thinking to our manufacturing processes and analytics, it's quite an enlightening approach and gives one an idea of just how much data one could obtain from the manufacturing process if you make the attempt to gather more information. Fortunately, as cell therapies become more widely used, as they enter the commercialization phase, and as they are increasingly automated, manufacturing processes and other aspects of the collection and delivery of cell therapy products tend to generate very, very large amounts of data. So automation and simply the large N's that go with commercialization give us the opportunity to capture much larger amounts of data about our manufacturing processes and other aspects of delivery of cell and gene therapy products, and also give us the opportunity to capture the data that we simply had not thought to capture in the past. And this is what I mean by taking a kind of a Google timeline or a Google approach in terms of capturing a broad range of data. And all of this, if analyzed effectively, can enable our understanding of critical process parameters and critical quality attributes. So this is what I would think of, these are the kinds of things that I would think of as big data solutions or potentially solutions. The ability to capture and to integrate and analyze a very broad range of data inputs and very much multidimensional data inputs from a wide variety of sources, not simply the QC testing results that you have, but a very wide range of data points related to the product, to the critical raw material, to the manufacturing process, to the final product. This requires both active real-time data acquisition as well as mining of data that's been acquired over time. And all of this is ideally suited to a big data approach. So cell therapy and gene therapy right now is at a very, very, is at a time where big data is a very opportune strategy. Big data allows capture and analysis and use of very large, often very complex data sets, and fosters data mining and advanced analysis with things like artificial intelligence and machine learning. Now, lest you think I'm making most of this up, and lest you think this is something that is half a century away or something like that, I'd like to point out a few examples in which big data approaches are being used now in cell therapy and gene therapy. One, manufacturing process optimization and control. A good example is Oxford Biomedica's collaboration with Microsoft to optimize the lentiviral vector production, to identify and track critical process parameters, and to modify their lentiviral vector manufacturing process to take advantage of what they've learned from data mining and from capturing real-time data and analyzing it in a very deep way. FDA is also taking big data approaches. This figure is taken from a recent FDA publication describing an FDA CBER project to develop a machine learning technique to identify populations of mesenchymal stem cells which, when stimulated, have immunosuppressive activity. And of course, immunosuppressive activity is one of the functions that MSCs are widely used for. So this is a very clever approach. The reference is given on the slide. It's to a cytotherapy paper and makes for very interesting reading indeed. Another example would be process management. And here the example that I have in mind is Vineti. This is a company that is developing and deploying data management systems to manage workflow and manage capacity, to link to operations at the clinical site, to link to manufacturing operations. You're going to hear a lot more about this, and indeed about many of the other topics that I've just mentioned, from Heidi Hagen from Vineti in just a few moments. A couple of other examples of places in which either big data or machine learning approaches are being applied. One approach that will facilitate a big data approach, that will facilitate big data analysis, is FDA's program to create databases for safety and manufacturing information about CAR T cell therapy products. Another is CancerLink, a project from the American Society for Clinical Oncology. This is their first big data project to share real-world data on oncology patients. And last, an NIH program for monitoring gene therapy protocols called GEMCRIS. GEMCRIS actually was deployed, I think, something like 10 years ago, and is an invaluable database for gene therapy. GEMCRIS is being closed down and is turning into another kind of a more modern kind of gene therapy monitoring database, but is an absolute gold mine for an absolute wealth of information about gene therapy products, about clinical outcomes, and to some extent about manufacturing. So all of these are either active big data approaches or are data sources for big data. And that leads me to my last, one of my last couple of topics. One is crowdsourcing data. Now, it's fairly clear that the data generated by one developer of a cell therapy, one company, can be very helpful. But the real value of big data comes when data is shared, and this is what I'm calling a crowdsourcing approach, when data is shared from different sources. Many companies are working on CAR T cells. Many companies are developing mesenchymal stem cells. There may indeed even be some points that can be, there may indeed be some critical information that can be gleaned from combinations of information from multiple types of cell therapies. But by sharing information in the pre-competitive space, it's possible to improve understanding of critical process parameters, of critical quality attributes, and so forth, for CAR T cells in general, or for MSCs in general, and these are just two examples. Now, this presents interoperability and software compatibility considerations. There are also a wide variety of data parameters that are measured. All of this adds complexity and requires standardization, which is my last big topic. To really make data sharing work, to make data mining work, to make machine learning work, will require real data standards. Crowdsourcing of data, that data sharing is actually a true lunatic possibility that's unrealistic. I point out that this is actually something that big pharma companies have been doing for years in the pre-competitive space. It's simply somewhat new to us in cell therapy. But as cell therapy and gene therapy grow up, and the commercial era of cell therapy and gene therapy continues to expand or continues to grow, I think we can find more and more opportunities to share data, again, in the pre-competitive space, as our big brothers and sisters in big pharma do. But to do that, it's going to require data standards, and that really is the last point that I wanted to cover. There are a number of existing data standards and standards that are in development for cell and gene therapy products. You see, and of course, we have benefited from a wealth of standards about other aspects of cell and gene therapy, whether it's standards for raw materials, standards for analytics and processing, from a variety of sources, from USP, from ISO, from ASTM, and of course, FDA has very much supported the development of standards. Data standards are simply the next step. And there are international collaborations to address standards for some elements of manufacturing and analytical data already in place. One that I think is worth emphasizing in particular is data standard C16 on data acquisition, and this is from ARM, the Alliance for Regenerative Medicine body, the SCB. So this is probably the most comprehensive data approach to generating data standards for cell and gene therapy products that I've yet seen. I won't read the quotes from the data standards that I've included on this slide, but I wanted to give you the opportunity to see them. But ARM essentially is saying much more concisely in these two paragraphs what I've been trying to get across for the last few minutes, that fundamentally the data that we generate throughout the development of cell therapy products and gene therapy products is an invaluable source of information about critical quality parameters, about critical quality attributes, and all of this facilitates efficacy, safety, and certainly supports regulatory compliance. But because the nature of data across from different sources varies greatly, we need standards to make it possible to share data, to support interoperability. So the objective of standard C16 is to implement protocols to increase the quality of data, to make it easier to share, and to enable mechanisms to share data while still safeguarding intellectual property and, of course, patient privacy. This proposed standard was published quite recently in the spring of 2019, but there's a working group already in place chugging away on this, and I would encourage you, if this is of interest, to participate in the C16 group. And with that, I'll be happy to turn over the presentation to Heidi Hagen from Vineti, who will go into greater depth about all of the points that I think all of the points that I've covered thus far. Heidi, take it away.
Thank you, Scott. And thank you for joining us today for this important, timely topic in cell and gene therapy, data management. My presentation will now shift to manufacturing and product handling within the value chain of cell and gene therapy. Before we dive into the slides, I would like to give you my background and share with you why I think this topic is so important to the cell and gene therapy industry. I've spent the last 30 years of my career in biotech, specifically the last 18 years in cell and gene therapy and personalized therapies. I was a senior vice president of operations at Dendreon for 10 years, responsible for the development and commercialization of Provenge, the first active cellular immunotherapy approved in 2010. It's manufactured at a volume of greater than 14,000 products per year, which is still the largest volume produced globally for any cell therapy. I was subsequently the global COO for Sodio in Prague, which included building a facility in China, manufacturing in US, Europe, and China. I currently sit on the board of 2 publicly traded cell therapy companies, Vericell and Zyopharm, and also a private manufacturing company, Lichen Biosciences. I am the co-founder and chief strategy officer at Vineti, which had been mentioned by Scott before. It is a cloud-based software company that manages the key activities and provides GMP workflows for all of the stakeholders in the cell and gene therapy ecosystem, from order to treatment. Our platform initiates and maintains the chain of identity and chain of custody while providing all of the stakeholders a transparent view of all the products, their status, ensures maintenance of the chain of identity and chain of custody. Let's start with a quick review of the current cell and gene therapy industry landscape. It was another growth year, and we now have more than 1,000 regenerative medicine therapies in the clinical development. Some examples are presented here for you. Kite Gilead shared their continued growth of one of the first CAR T products, Yescarta. Bluebird received an MAA approval in Europe for the first gene-modified cell therapy treating beta thalassemia. And we are continuing to see mergers and acquisition and consolidation in the space as these products mature and are validated through regulatory approvals. Despite the great news and growth in this industry, we continue to have challenges, especially in data management. The science behind cell and gene therapy and personalized therapies has moved forward rapidly, data capture and monitoring that support the science. We have data silos that are not integrated. Our alert systems are not based on sophisticated analytics such as AI, and such mistakes and they limit appropriate interventions and improvements as needed. And this can all result in lost or failed products, repeat work, and higher costs than necessary, both for the operations and preparing the data package for your regulatory submissions. And in addition to these data challenges, we need to consider the range of personalized therapies that are currently in development. These different product categories presented here create different data and data management complexities throughout the supply chain and in manufacturing. Here we have outlined 3 major product categories to illustrate some of the complexities. For example, in gene therapies, the data includes individual patient genetics, laboratory information management system integrations, and basic chain of identity and chain of custody linked to patient registries. In allogeneic therapies, these therapies are manufactured both as one-to-many and one-to-one type products, similar to autologous. They require data management of multiple chain of identity and chain of custody with donors involved, which is a look-back, look-forward type system. It also involves HLA mapping and inventory mapping and logistics progress data throughout the chain. For autologous, these therapies are the most complex and data intensive. They require complete data capture and management from order to treatment, including chain of identity and chain of custody to ensure product quality and patient safety. This is a picture of the autologous journey and the stakeholders that are capturing valuable data. An allogeneic journey is somewhat different but similar in a lot of ways. Per arm, the current open trials can enroll up to 50. Each of these links in the chain are varied and are in different systems and can sometimes involve large databases, as Scott had discussed before. Compiling, analyzing, and reporting the data from these product journeys is critical to maintaining patient safety, gaining better understanding of how these therapies are working or perhaps not working, and providing these therapies to all patients in need, regardless of their location. The complexity in these therapies, combined with the industry growth, requires us to industrialize data management. The ordering and scheduling needs to be standardized, easy to use, and it must be able to integrate into the already existing healthcare processes. By having intuitive interfaces, integrating with legacy EHR, electronic health record systems for data exchange, providing simple GMP work instructions for data capture and flow, and enabling data exchange with hubs and other supporting systems in the value chain. Manufacturing systems need to provide real-time data from the supply chain and the shop floor. Flexible facility loading, rescheduling of patients as needed for those real-time world events that we all know occur, and enabling real-time troubleshooting with targeted operational interventions that are effective. These therapies, because of their biological and genetic modifying nature, require that data monitoring analysis be available and transparent throughout the complete value chain. The data must also be secure and used only by those authorized to do so, ensuring that the supply chain is reliable and robust. Now we will drop down one level and look at the types of data that I referred to on the previous slide. I will not go through each of the bullets, but rather discuss why the accurate and timely capture of this data is so important. For the order process, the critical patient identifiers are necessary for the creation of the chain of identity. The prescribing physician information and specific medical center delivery data are captured at this point in the value chain and must be accurate to ensure patient safety and proper delivery of the product at the end. In schedule with open cell collection slots, courier pickups and drop-offs, open available manufacturing slots, and a calendar linked to other concomitant therapy schedules requires real-time access to calendars, resources, and logistical portals across the complete chain. In the collection process, this is a GMP process done in medical centers every day. There must be a clear and secure way to ensure that the patient identifiers match the chain of identity, and all the critical data throughout the collection process is captured in a standard format, thus enabling the data to be loaded into a report that can be used as the start of the batch record. There must be an easy and secure way to capture the data and create a GMP record in an environment that is not typical GMP contractor environment. In manufacturing, regardless of the length of time of the manufacturing process, the chain of identity and chain of custody must be maintained and the critical operational data points captured to ensure a complete operational control and enable progress and chain of identity to be tracked throughout. And at the infusion point, proof of delivery is a legal data requirement. And most importantly, the patient must be matched to the chain of identity and treated with their product and not someone else's. So I took you through the drivers and reasons for having good data management in a real-time and accessible manner for personalized therapies. The FDA and EMA and other regulatory authorities have begun to codify their expectations around data management and traceability for these therapies in their guidance documents and directives. I invite all of our listeners to locate this document online and read through it if you have not already done so. Data management beyond the already prescribed GMP documentation practices is becoming key to maintaining compliance to current GMPs. Like the FDA, the EMA published new directives for ATMPs. And in fact, I would suggest you read this because the EMA was even more prescriptive in their expectations, and it's a very helpful guide. Like Scott, I'd like to discuss a bit about the standards coordinating body. In addition to recognizing the need for data industrialization and modernization in these therapies, the FDA funded a standards coordinating body, which is made up of volunteers from the industry. And I would suggest if you are interested in the participation of this, we welcome you to go to the URL on this slide and find out more information. Establishing standards needs to be aligned with the guidance documents and directives. There's a large amount of data in the cell and gene therapy ecosystem, as Scott discussed, that needs to be captured and effectively utilized for the sake of patient safety, expansion and use of these therapies beyond a limited number of just a few medical centers, and creation of new and more effective therapies down the line. Now, I'm going to go through 6 strategies I'd like to suggest for successful data management that are important for anyone involved in developing these groundbreaking therapies. Strategy number 1, automate. Focus on automating critical process steps and data elements for the end-to-end process that protect patient safety, support efficient operations, and streamline the path to filing. This delivers data completeness and standardization, improves efficiency and quality, and can reduce your resource needs. It also provides data availability for real-time status, alerts, and reports. Suggest leverage cloud technologies to maintain compliance across the distributed supply chain, and use intelligent algorithms to optimize regional scenarios, capacity management, and scheduling across all activities, including collection, manufacturing, transportation, and treatment. Some people consider automation as removing manual product manipulations and human labor from the process. This is one type of automation, but it can also mean using digital platforms to assist in processing and data capture. In cell and gene therapy, data capture is required outside of the traditional manufacturing process that is done within the 4 walls of the GMP facility. Automating the workflow and data capture is critical not just to ensure accuracy and patient safety. It reduces the cost of the supply chain, as noted by Simon Jacobson in this publication snippet above. For example, automation can yield a projected 25% reduction in rejects and reviews that would be done manually without the data automation. Strategy number 2, adopt technology early. Implementing a digital platform at the start of a trial or trial phase instead of doing it mid-trial or mid-phase can save you time and money by avoiding retraining, conversion of existing data, and change management. Each product in this process will drive when digital data capture is best to implement. Some products have so many moving parts and participants in different locations that even small numbers in production require a good digital data capture system. As the clinical production volume grows, it is easier, cleaner, and less expensive to have the system ready so that in later phases, when you are capturing registration data for your BLA or MAA submissions, you can demonstrate a safe, robust, and effective supply chain. As noted in the previously published white paper that I co-authored on data management, these advanced products are driving different developmental cycles. The change in activities with patient-specific data through the clinical phases necessitates that the data be ready, available, and analyzed. Without standardized data available in real time, your development timelines will be delayed and your data quality will be lower. Strategy number 3, employ ecosystem expertise. Choose digital platforms that demonstrate prior regulatory authority acceptance and perform them with full-scale operations. Experienced resources are limited in this new evolving space, I think, as we all know. Look to ecosystem partners for expertise in critical areas such as technology and a deep knowledge of software practices in a regulated environment, and operations and regulatory experience in real-time biological supply chains. Support operations and compliance with proven digital platforms, and leverage off-the-shelf configurable software versus one-off custom developed for simpler updates, regulatory familiarity, and support of the industry standards that we talked about before. This diagram outlines how the supply chain of a cell and gene therapy product has many data sources that must be integrated in a secure manner that the regulatory authorities understand and trust. Across the whole ecosystem, you need to be sure to select systems and partners that have a deep understanding of how all this data needs to be standardized, formatted, and compiled to enable reliable, safe, and robust production and regulatory submissions. Naive partners can hinder your regulatory approval and can cost you legally if the system is not secure and validated for your defined processes and product. Strategy number 4, which we talked about a lot before, but I can't emphasize it enough, is to standardize. Work with all stakeholders, internal and external, on standardized data collection and utilize systems that make standardization as easy as possible. Early data standardization ensures that data are readily available, accurate, easily analyzed, and interpreted appropriately. Data standards do not exist broadly, as we've discussed in this industry for cell and gene therapy, and can differ from organization to organization. Data variability comes from disparate data collection and reporting systems in the ecosystem, which include paper, digital, and then a mix of both. Standardizing data and processes is a significant part of creating a complete and easily analyzed data set. And a complete data set is important for patient safety, which I cannot underscore enough, understanding the product and the process, and optimizing the overall patient experience. These visuals give you a picture of the before and after for a centralized and standardized data management system implementation. As we fondly call the left the spaghetti diagram, you can see how many manual and sometimes incongruous digital systems can cause a lot of back and forth and sideways information and data flow, where no one has a single one-stop shop for input and extraction of data. The right is where you should set your sights, a flywheel opportunity where all participants in this critical supply chain can have a single point of data and can easily have a transparent view of the product and its progress throughout the chain. Strategy number 5, make collaboration manageable. Implementing digital collaboration early delivers benefits upon implementation and enables easy and low-cost scaling up and out. It also lowers the barrier to adoption of new technologies such as blockchain and machine learning later on. The ecosystem contains many partners, functions, process steps, and geographical locations that require effective collaboration for data capture, patient safety, and successful treatment. These many-to-many requirements are best supported digitally, and this approach provides the following benefits: real-time compliance, patient safety, and actionable information, seamless, efficient, and low-cost scaling up, and reduces time and effort to gather and synthesize multi-stakeholder data. Anyone who has run a biological production operation knows information on each unit operation and its health within the defined tolerances is key to being in control of the supply chain. With live cells as the backbone of this production process and the final product, real-time information is critical to being able to monitor progress and to intervene with reasonable action as required. Since a significant portion of this live cell supply chain is handled by personnel and entities outside of the GMP manufacturing facility, real-time visibility combined with the previously discussed standardized workflows and data are key to ensure that the product is safe, meets quality parameters, and most importantly, will be delivered on time to the correct patient. As we just discussed, there's a need to orchestrate and collate all supply chain data across multiple entities. And we must also have an ability to integrate with the different layers of the system within the manufacturing facility and other locations like cell collection. The diagram here on the right provides examples of data sources and systems of record that capture the primary data. By integrating the data sources, a complete data-driven account of the individual and multiple products in a pipeline can be viewed, and proper operational actions can be applied as required throughout the chain. Strategy number 6, optimize your analysis and reporting. Stay ahead of those regulatory requirements we've discussed for long-term patient monitoring by laying the groundwork now. Start with asking the relevant focused questions and begin gathering intelligence throughout your chain and throughout your process. A high-quality data set helps ensure that the information is readily available for that analysis and reporting. Real-time reporting and data analysis are critical for ongoing operations and optimization of that with compliance, especially as patient volumes increase. Provides insight into the product, process, and performance, and can be enhanced by machine learning and artificial intelligence, which may lead to valuable additional discoveries. And also enables long-term follow-up requirement, as many of these advanced therapy approvals now require 15 years and likely will be important to do it longer than that. Here are a few examples of dashboards that can be viewed and used by multiple stakeholders, including healthcare providers, therapy company executives, supply chain managers, and shop floor schedulers. In summary, these are the 6 strategies for success that I recommend. Strategy 1, automate. Strategy 2, adopt that technology early. Strategy 3, employ ecosystem expertise. Strategy 4, standardize. Strategy 5, make collaboration manageable. Strategy 6, optimize your analysis and reporting. Proactive adoption of these data strategies is essential to harnessing big data in cell therapy and driving more efficient, reliable, predictable, cost-effective manufacturing. Taking those 6 strategies in combination with the ATMP landscape and the updated regulatory guidance we discussed at the beginning of the presentation, this view of the value chain shows you how an integrated ecosystem can provide the modern data management required to successfully order, schedule, manufacture, and treat the patients in need of these life-saving products. Even early clinical trial products have data sources you can see at each link in the supply chain at the top of this diagram. If you do not yet have a data integrator like Vineti pulling these data sources together, there are existing manual processes and individual data sets like Excel or clinical trial databases. It's important to look at your development, timeline, phases, and understand when complexity combined with your product volume will necessitate bringing in a holistic data standardization and a management system like Vineti. And in fact, the earlier that this is done, it is to implement and will limit retraining of all stakeholders. An increasing number of these stakeholders are already trained on this system and others as the number of the products grow, and they prefer to have a standard approach versus several one-off manual approaches. A data solution such as this enables simplicity and scale. Real-time data capture and access enables 24/7 management, which is required, of trials and customer service. Metrics on current operation enables more successful troubleshooting and management tactics as things happen. Data tracking and trending at scale enables predictive operations such as seasonal trends or apheresis reschedules. Transparency of data and product journey progress helps build trust with healthcare providers, patients, and others within the value chain. And an integrated patient logistics, product conditions, and product data enables better product characterization, similar to what Scott was discussing in his presentation. The ultimate goal here with modern data management is to optimize your supply chain while providing safe and effective products to the patient. Safety is the backbone of your brand and your product, so optimize your big data. Participating stakeholders are in different entities and geographies. Transparency and ease of access is key to all of those involved with it. Each stakeholder has a different role in the journey, so the data must be useful for them. Management of the product journey and operation needs real-time data for real-time action. On-premise or legacy systems do exist, and they must be integrated for a single, more complex and get greater volume. Privacy and role-based access are also key. Regulations will always rule in this space for a good reason. Here's my last point. Your big data analysis and usage can only be as good as the data itself. So plan your data management strategy early and in ways that will enable growth and scale of your product and your process. Thank you. And I will hand it back to our moderator, Stephen, for question and answer period.
Thank you for that presentation, Heidi. Before we begin the question and answer segment, once again, I would like to remind our audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, what is the single biggest obstacle to adopting a big data approach in the cell and gene therapy industry today? How feasible is any of this for a cell/gene therapy startup? And that question is for you, Dr. Berger.
Thank you. Well, first off, before I answer any questions, it occurs to me that I should make clear I am a consultant in cell therapy and gene therapy, but I have no relationship with Vineti at all. Other than that, I think what Vineti is doing is important in advancing the field. So full disclosure, I have no biases with regard to Vineti or relationship. On to the question. Single biggest obstacle, candidly, I think the single biggest obstacle to adopting a big data strategy is the idea that it is too difficult to do early on. This, I think, is very much a fallacy. I think the biggest obstacle is really more one of the perception than one of Heidi brought out. It's important to turn to a big data approach as early as possible. It's important to begin capturing data since this will be an invaluable resource throughout development and throughout phase 4 commercialization. So I think the single biggest obstacle is actually the idea that somehow this is too difficult or too far in the future or unrealistic. I think one of the, just as we build our strategies for supply chain and just as we build our process development plans, so too from the very outset do we need to build our data management plans. And I think if we do that, and if we support standardization of data and development of data standards, I think we'll find in short order that big data begins to pay off for us.
Thank you. Our next question is, with limited budgets and resources and the added pressure to keep clinical trials progressing as quickly as possible, where is the best place to start? What is the single highest impact or ROI thing I can do? And that question is for you, Heidi.
I think probably the best place to start is really to map out your process and to really understand who's involved in your supply chain from order to delivery. And then additionally, once you've mapped that, is to really go in and look at all your data sources. Realistically, you have to know what you're dealing with before you do anything else. And by doing so, even if your choice is to automate certain parts and not automate others, you now have a good understanding of what's going on with your product. You've if you will, characterized the bigger, broader process. And so I think that's probably the most important thing to start with, because then from there, you're able to develop what your user requirements are, who's going to be involved, what the system needs to look like, and what's most important based on the complexity and volume of your product going through that now defined process. So I think that's really the best first place to start, and then you can move to other steps later.
Great. Our next question is, how do you know where the tipping point is for the complexity by volume that necessitates implementation of an automated digital platform? And what does it look like when you are approaching that point? And that point is for you again, Heidi.
I think this kind of pulls a little bit off of the question and answer I just provided, and that is you can have processes and products in this environment that involve, say, genetic information plus capture of cell collection like an apheresis, plus you actually may even have some tissue that you excise from a patient. So you can have even just a small volume of patients going through, say, an early phase trial, but the complexity of the process, which may involve multiple streams of information and physical product, needs to come together at some point in manufacturing and or delivery back to the patient. And so I would say it's really looking at your individual process and product. How many different stakeholders are involved? Where is the geography? How many clinical sites are you working with? And that will define for you that complexity piece. And then as you take into effect what the volume is, are you in phase 1, phase 2, multiple phase 2s, or a very large phase 3? I think you have to look at and think about patient safety being number 1. Number 2, on top of patient safety, which is just another part of patient safety, is understanding your process and your product so that you can really truly have a robust, reliable process. Is you need to look at how do I take those two aspects of complexity and volume, and at what point can I have comfort that the patient is safe and that we also can capture all of the data so we understand the product better. And it's going to be an individual situational question, because it will be based on your product, where you are globally with it, and then combining that with the complexity and the volume. And so I think it's something that it's kind of a analysis that's done internally for the company, and recognizing at what point aren't we comfortable that we're going to cover all of this. But back to what I think Scott had said in his presentation is the data you need to capture early so that you can really start to build that database. So it would be a question of when can you afford it, tipped with the complexity and volume.
Great, thank you for that answer. Our next question. Is machine learning / AI really a practical tool at this point, or is it mainly for academic programs and large cell and gene therapy companies? And that question is for you, Scott.
Well, I think one of the example slides that I showed attempt to answer that question. Certainly, I think this is a practical reality. A couple of the examples are taken directly from industry, but it's also, I think, worth bearing in mind that there are several cell therapy and cell therapy related companies today that are not just using machine learning, but indeed are built on machine learning platforms. There are cellular immunotherapy companies that have at their very foundation the ability to use machine learning to identify suitable neoantigens, for example, for cellular immunotherapy. I believe Gritstone is working on that, and I'm sure they're not the only one. So it's very much, I think, a practical reality. It speaks volumes, I think, when you see that FDA is working on or has developed a machine learning technique that's predictive of optimally immunosuppressive mesenchymal stem cells. So this is not something that's 22nd century. This is something that is very much a practical reality here and now.
Great. Our next question is, how could crowdsourcing data work? Why would cell and gene therapy companies be willing to share data? And that question is again for you, Scott.
Well, I think how it works, well, it will work better and better as data standards become more solidly established and widely applied. To the question of why would companies share their precious process information, again, this is something that the pharma industry went through years and years ago, and has found ways to share data in the pre-competitive space. One of the things that I think is particularly valuable about the STEAM project is that it includes defining data standards that allow sharing of information without impairing or limiting the company's IP, the company that shares the data. So it is certainly possible to do this. Pharma has been doing this quite literally for years. And the motivation is a simple one. You can glean more information based on a larger N of data. And just because two companies might be working on a CAR T-cell therapy doesn't come to both companies to share certain aspects of their data, as long as their IP is protected and as long as patient privacy is assured. Those are entirely possible and feasible. And this is a situation in which sharing really does result in a net gain, or can result in a net gain for all involved.
Okay, great. Our next question. I understand the need to implement a digital platform to capture data, especially to ensure traceability and patient safety. But other stakeholder groups / functional areas in my organization don't. How can I convince them? And that question is for you, Heidi.
I think all stakeholder groups and different departments and functions within a therapy company, and really walk through what's involved with order through to delivery of these products, of their products specifically, I would say, and really walking through a little bit of, we discussed mapping the process, but as walking through all the stakeholders, showing where all the data is. But then really, honestly, I think it comes down to is really everybody should recognize that this is for the benefit of the patient and, most importantly, the patient safety. And that's when you think about like chain of identity and chain of custody and being able to pull up all of that data and be able to have a very transparent system that enables all stakeholders to be able to see where is that product, who does it belong to, and what are the attributes associated with that product, and where is its progress. And I think when you walk through that as a cross-functional group and you understand how critical patient safety is in this particular niche of biotech, and it's now no longer a niche, it's actually a very, very large expanding piece. And I think that's another piece also to bring into it, is to show, okay, we're not just the only cell therapy product, say, in a particular set of clinical sites. There's multiple cell therapy products there. And so in addition to taking your process, showing that, and really understanding what it takes to make sure that patient's safe and you're capturing all the right data, then you think about your product combined with others. And how do you make sure that you can do right by the patient? And I think that that would be the piece that I would emphasize is how are we going to do right by the patient? How are we going to capture all this data, which is going to support our product brand? And sometimes when you have folks that aren't closest to the technical operations or to the clinical trial, that's something that they don't think about on an average daily basis. And so I think when you give an explanation with facts behind it, I think it really helps support the need and the effort to put in a good data capture system.
Thank you. Our next question is, on the concept of interoperable and accessible data formats, how or are equipment manufacturers opening up raw data streams from their equipment to enable the integration of MFG equipment into an automated data environment? If not, what can the industry do to pressure them to enable this? And that question is, Scott, do you want to answer this first?
Sure, although I'm quite sure that Heidi will have a great deal to add on it as well. Well, what are equipment manufacturers doing? It depends on the kind of equipment. Clinical cell counters, for example, have had data outputs intended to go to hospital information systems and laboratory information systems, LISs, for, well, decades now. For cell therapy specific hardware, the answer is it depends. Some of the automated processing technology currently on the market or in late stage development does indeed have the ability to output data, but generally speaking, it's not necessarily output that's in a shareable cross-platform format. Oftentimes, it's in the form of a lovely report that that's GMP compliant, but isn't necessarily something that you can load into a database. The answer really is it depends. It depends on the kind of equipment that you're talking about, and it depends in some cases on how long that piece of equipment has been around and what its legacy is. As I say, for some kinds of equipment like cell counters, there have long been, for many cell counters, there have long been data output ports. Those aren't necessarily data output ports that are in a format that's the most modern or usable. And some older pieces of processing hardware that are commonly in use do not have data outputs at all. But more fundamentally, what can the industry do to pressure companies to enable this kind of capability? Well, I don't think a whole lot of pressure is needed. I think all you really have to do is consistently build this into your user requirements when you're looking for equipment, and make sure that companies that are developing technology, that whether it's process technology or analytical technology, make sure that the companies that are developing this kind of technology know that this is something that you're going to have in your user requirements. Also, I would encourage you to participate in any of the several automation working groups that currently exist and that welcome new participants. Most of all, most fundamentally of all, make sure that the technology companies that support cell therapy through process manufacturing equipment and analytical equipment know that this is something that is going to be a user requirement, and there really are no two ways about it. It's not a user preference, it's a requirement.
And Heidi, do you have anything you'd like to add?
No, I think that was a great answer to it, and I would emphasize, I think the great point there with user requirements is really making it known that this is something that's required by you as a customer of these manufacturing equipment providers. And yes, I find that when you look at the two categories, QC, analytical equipment, and hardware, it tends to have an API or an integration point that works well for pulling and pushing the data, whereas I think we either have it very new or very old, and it doesn't necessarily either end of that spectrum have that. So I think it has to be something that's emphasized from the customers buying this, and it's through the user requirements that, you know, they'll either be accepted in an RFP process or through the development process, and I think that that's really the best way to do it. So I support that approach also.
Well, thank you both for your question answers, and thank you audience members for your questions. Before we finish the webinar, I would like to ask our presenters, Scott and Heidi, if they have any closing remarks. So, Scott, do you have any closing remarks for our audience?
Just this, I actually just saw a question pop up asking if there's an integration data capture strategy for patient self-reporting, for example, by phone. I'll make my closing remark an attempt to answer this question. I do not think that there is such a strategy. Heidi may be aware of one, but I think it's an excellent point, particularly with regard to long-term follow-up for adverse events for patients receiving gene therapy products. Long-term follow-up, I think, as Heidi mentioned, can be 15 or 30 years for all practical purposes, really for the rest of the patient's life. That's for gene therapy, for patients receiving gene therapy products. There are many circumstances, however, in which you would want long-term follow-up, or at least some level of longish-term follow-up for patients receiving just cell therapy products. And patient self-reporting by phone, I think, is one very good way to do it. So would an online format. I think it would be imperative to make sure that you have suitable means of protecting the patient's identity, privacy considerations. But I also think it would be essential to make sure that you can unambiguously identify the patient. Fortunately, the banking industry has established this very, very well with things like not just unique sign-ins with passwords, but tokens such that you have to verify your identity by putting in a cell phone-based token that has a finite lifespan. There are ways of solving that problem. To my knowledge, they aren't being applied yet in gene therapy and cell therapy. I may be mistaken about that. But I think that's certainly something that needs to be considered and can be very, very valuable in terms of patient follow-up. And I'll let that stand as my closing remark and hand it off to Heidi.
And I'll just make a quick comment on that, is that we're not aware either from a Vineti perspective on patient self-reporting opportunities. However, we do get the requests from almost all of our customers, especially those in phase 3 heading into commercial and or in commercial, is to have a patient-facing app such that there can be, as we just discussed, the ability to have the buy-in, have the security and everything associated with that, but then have them be able to provide feedback and or push information to them to be somewhere they need to prepare themselves in some way, shape, or form, like drink lots of water and, have calcium-based food like yogurt, et cetera, which is the, kind of standard as you head into an apheresis. So we find it for patient education and for self-reporting is it's becoming something that a lot of companies want, but it's not there yet. So I think it's something we will see come along here soon. And just in closing, I would say these advanced therapies are exciting and promising. The segment is experiencing significant growth and innovation, but we haven't really modernized our use of technology and data management yet, as you've kind of heard us discuss today. The increased complexity and data management requirements of these therapies have really, as we discussed, prompted the FDA and the EMA to issue, the guidance at changing this. And, the products I've overseen have supplied, more than 100,000 cell therapy doses in early clinical phase trial to commercial. And so I want to tell you from my perspective, the priority is to ensure patient safety. And in doing so, companies will benefit more from, these compliant and efficient operations that also come from these, having good data capture and a streamlined regulatory process with the product. So I'd say now is the time for companies to really get ahead of some of these requirements and simplify their operations and really look to the 6 strategies that we outlined here in the presentation and take advantage of the new platforms that have emerged really to solve, the data management challenges that are specific to these advanced therapies in the manufacturing environment that they're in. And I want to thank everybody for taking the time to listen to us today. It's been a pleasure, and it's a really important topic, so we're happy everybody's really interested in it.
And I would like to take this opportunity to thank our presenters once again. That was Scott Berger, MD, Principal of Advanced Cell and Gene Therapy, LLC, and Heidi Hagen, Co-founder and Chief Strategy Officer for Vineti, Inc. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.